Hi, everyone. Thanks for joining. We're going to be getting started in a few minutes. In the meantime, just hang out. Just quick housekeeping. There is a little Q&A button at the bottom. If you have any questions throughout the event, feel free to drop them there and our team will answer them. In a few minutes, I will also launch a poll to kind of help us get to know who's in the room with us today. Yeah, so look out for that in a few minutes. All right, I'm going to be launching the poll. So please answer these questions to help us get to know you. And then we'll be starting 3 minutes after the hour. All right, 1 more minute for the poll, and then we'll get started. And like I said, if you have any questions throughout the event, leave them in the question and answer below, and our team will be happy to help you.
Okay, let's get started. I'm going to go ahead and share my screen. All right, well, I'd like to thank everyone for joining this morning for the Genomics Data and AI workshop. We're just going to go through the agenda before we get started. So we're going to start with a brief overview of how you can accelerate genomics data discovery with our unified data analytics platform for genomics. Then we're going to dive into a fireside chat with Dr. Evan Maxwell, who's a senior manager for data engineering at the Regeneron Genetics Center. We'll have a brief break and then go into the workshops. So there's 3 areas that we're going to cover. The first is on how to accelerate variant analysis using Delta Lake for storing data, for storing genomics data, and Apache Spark for processing it on a massive scale. The second workshop, we'll talk about how to do statistical genetics at scale using the open source project Glow that we developed at Databricks. We'll have another short break and then go into the final workshop, which will talk about Glow whole genome regression, which is an ultra scalable method for doing population scale genome-wide association studies or whole genome regression. And then we'll end with Q&A. All right, so at Databricks, the mission of the company is to help data teams solve the world's toughest problems. And there's no more difficult problem than analyzing population scale genomics data. So we're going to talk about why we're doing this, why it's such a big problem, why it's important, and then how we are solving these problems on a massive scale with companies such as Regeneron. Cool, so I'd like to start with a story of Nick Volker, who's a 6-year-old child who had severe inflammatory bowel condition that was unexplained. His parents spent a 3-year diagnostic odyssey trying to understand what the cause of his disease was. And he had over 100 surgeries. And in the end, after 3 years, they gave him this radical new technology called exome sequencing to try and find out what the cause of the disease was. And they uncovered it. It was a mutation in a gene on the X chromosome. And once they knew the cause of his disease, it actually immediately pointed to a treatment, which in this case was a bone marrow transplant. And he recovered and no longer had to have all these surgeries. So this is the power that genomics data analysis can bring to the healthcare industry. And it's so powerful that Francis Collins, the director of the National Institute of Health, wrote about this in the journal Science, and this was 10 years ago. So this technology was the first proof of concept for it was in 2009, and Francis Collins wrote about it here in 2010 as going to be absolutely transformative to the healthcare industry. So understanding the cause of disease is the first step to a cure. And it's not just Nick Volker who's been suffering with a rare genetic disease. 30 million people in the US live with rare genetic diseases. It affects 1 in 10 Americans. There are 7,000 different types of rare genetic disorder, 95% of which have no FDA-approved treatment. And another genetic disorder is cancer, and 1 in 3 people will contract cancer in their lifetime, and 1 in 4 people will die from the disease. And it is the number one cause of death among children. And it's just a really serious problem. And so being able to diagnose these genetic diseases is absolutely true. It's going to be absolutely transformative to the healthcare industry. So not only diagnosing diseases, but identifying treatments. So we know that pharmaceutical companies, 90% of experimental medicines fail during clinical trials. And pharmaceutical companies spend over $100 billion on R&D or research and development every year, and only 10 to 20 new drugs are approved. And when they look retrospectively at drugs that have been approved, now that we have this genome sequencing technology, it turns out that they're twice as likely to be approved if they have supporting genetic evidence. So incorporating genetic evidence is going to double just by knowing that a disease has a genetic origin, and knowing the genetic origin of that disease increases the likelihood of a drug being approved. And this is why national governments like the UK Biobank, the Genome Asia 100K project are starting to collect massive population scale genetic data sets to help realize this potential for healthcare. And the Regeneron Genetics Center has sequenced now 1 million exomes. So the first proof of concept was in 2009, the first application 2010, and now we have 1 million in 2020. So the rate of progress has been absolutely incredible to witness over the last decade. And it's not just diagnosing genetic diseases, but if you can link genomics data to healthcare data, you can actually predict the outcome of clinical trials. So you can improve the efficiency of clinical trials radically. So on the left is an example of a randomized controlled trial for selenium supplementation with prostate cancer. So there was a theory that selenium supplementation would reduce your risk of prostate cancer. And so they did a randomized controlled trial for selenium supplementation versus a placebo. And the randomized control trial controls for any confounders between the placebo group and the exposed group, and they found no effect. But there are also genetic variants that affect the level of selenium in your blood. So you can actually use genetic data. Instead of doing a randomized controlled trial, you can randomize individuals based on their genetic profiles. So you compare people who have a high genetic or ability to absorb selenium versus people who do not. And again, you can actually conduct randomized controlled trials using genetic data to randomize individuals. And in this case, they confirmed that the prostate cancer risk was unchanged by the exposure to high selenium. And so this was a failed randomized controlled trial that cost $114 million. And with genetic data, you can just show that you could show in advance without conducting the trial that this was going to fail. So linking genomics data to healthcare data can predict the outcome of clinical trials. It can also be used to conduct clinical trials. So here's an example, and this is for drug repurposing. So there are two lines here. So one is, so on the x-axis, we have your LDL cholesterol levels in the blood. And on the y-axis, we have your risk for coronary heart disease. So if you have higher LDL cholesterol in your blood, you actually have a lower risk of coronary heart disease. And there are genetic variants that associate with this in two specific genes. So in these two genes, if you have the genetic variants that lower your LDL cholesterol, you have a lower risk of coronary heart disease. And there are drugs that influence these two genes. The genes are called HMGCR and NPC1L1. But there are drugs that target these genes, or the proteins in these genes, and also lower LDL cholesterol. So the genetic variants lower LDL cholesterol, and the drugs do the same thing. And these two drugs are called ezetimibe and statins. And this other line shows the randomized controlled trials that were done and the effect of those. So the genetic data, it's very clear that this combination of effects of these two genes will reduce your risk significantly. And the randomized controlled trial shows it too. So using genetic data, when you know the genetic mechanism, and you know the mechanism by which the drug acts on specific genes, you can conduct randomized controlled trials for drug combination therapy and also drug repurposing for different conditions. So the data itself can be used to conduct a randomized controlled trial without having to go through the expense of having people come in for the trial. You still need the trials, but you can tell in advance whether or not it's going to work, or you can increase the likelihood that it will work. So this population scale data is worth billions of dollars a year to the healthcare industry. So there's two areas: the drug repurposing. So you can identify new drug indications, so new ways that drugs can influence other diseases. And that's 25 to 40% of pharmaceutical revenue today. And you can reduce the failure rate for clinical trials, which can cost $100 million each. So drug repurposing, it's transformative in that area. It's also transformative in R&D for drug discovery. And these companies are spending over $100 million collectively a year on R&D. So you can reprioritize R&D and you can repurpose existing treatments. And the ultimate goal is what we call precision medicine. So treatments based on your individual genetic profile and your individual lifestyle and environmental exposures. And this is only just starting to unfold. There are new treatments that are coming out for rare diseases and cancer. It's a trickle today. It's going to be a flood in the next decade. And for existing treatments, you can identify, based on your genetic risk profile, the optimal drug dosage, the optimal drug combinations, and you can minimize side effects. So your genetic profile will also, you can weigh the risks and benefits of taking specific treatments by combining the genetic data with your healthcare data. So absolutely transformative, but it's a massive problem. So genomics is a real big data problem. Your genome is 6 billion letters long, or 2 copies of 3 billion each. And but the cost of sequencing a genome has dropped dramatically, and because of the massive investment from the Human Genome Project 30 years ago. So now the cost has dropped to under $1,000 per genome, which has led to an explosion of genetic data. And it's predicted by 2025 to even dwarf the data that's being generated by the likes of Twitter and YouTube and the entire field of astronomy. So genomics data is a massive big data problem. And when you reach population scales with genetic data analysis, or what's called bioinformatics, you encounter a number of challenges. So the first is the data is siloed and messy. So you have the healthcare data on one side, where there's specific tools for working with that and specific file formats. And then you have the genetic data on the other side, which again has custom tools, custom data formats, custom ways of analyzing the data. So harmonizing those two, harmonizing each of them individually is incredibly challenging, but then bringing them together is an even bigger challenge. The second is, yes, so this is related to the analytics. In the case of bioinformatics, you have custom academic tools that are run from the command line. They're not well maintained. They're very difficult to use. They require highly specialized skill sets to work with. And then you have disconnected teams. So you have researchers and clinicians who understand healthcare data. You've got computational biologists and bioinformatics who work in silos. They write code in different languages. They use different tools. They have different skill sets. And enabling these teams to work together is critical to accelerating this process. So this is why we've created the Unified Data Analytics platform for genomics to enable interactive genomic data processing and analytics at scale. And there's 3 areas that we are focusing on. So first is being able to unify the data. So breaking down the silos between these different data sources, be it genomics data, be it data from the academic literature, or medical billing claims codes and things like that, electronic health records. Being able to unify all of those different data sources and integrate them using machine learning. And we're going to talk a little bit about Delta Lake for storing that data. Delta Lake is like it's been called like the AI database. So it's a way of adding a structure and schema to semi-structured and unstructured data on a massive scale. Second is analytics. So Databricks, we've built a very powerful platform for doing data analytics in the cloud. And the demos will highlight how the platform can be used to do data analytics. And the third is collaborative workspaces. So the Databricks platform enables teams to work together. You can think of it as like it's like an IDE or an IDE for doing data science, and combined with Google Docs. So you can have teams working together interactively in real time on code and remotely working together and to get more productive, to avoid reinventing the wheel, to learn from each other, and to get things done much faster. So unifying the data, providing powerful analytics to scale up, and collaborative workspaces for teams to improve their productivity. And to dive a little bit deeper into what we're going to talk about today, we've developed this framework called Project Glow, an open source toolkit for doing genomics tertiary analysis or genomics data analytics on mutation data or variant data. And there's three areas where you can use Databricks to process genomics data. So the first is the raw data processing, and that's from these raw FASTQ files that come off Illumina sequencing machines, get processed to BAM and then to VCF. So we've developed pipelines to do this seamlessly and in a highly accelerated way. And then once you have the variant data, though, this is where Spark really comes in for just scaling up the processing of data across many samples. So we've taken the open source G80K toolset for doing joint genotyping and used Spark to parallelize this on a population scale. And Karen's going to talk about that later. This is called joint genotyping, or merging of massive numbers of variant call format files, which can then be stored in Delta Lake, which sits on top of commodity cloud storage. And Delta Lake will basically provides a schema to or structure to these VCF files and allows you to do scalable analytics on them. Amir is going to talk about the quality control and statistical genetics steps that you would do once you've merged the data. And then Kiyavash is going to talk about how to do whole genome regression at scale. So associating or linking genetic variants with the clinical data. So this is the process. And in Databricks, we've also incorporated open source tools such as Hail for doing this analysis, and we can go back and forth between Hail and Spark and Delta or Glow. So we can seamlessly go back and forth between existing bioinformatics tool sets and robustly engineer this pipeline to go end to end. So, and then finally, the collaborative workspace environment has a number of different tools for data scientists or bioinformatics to push the research forward. So we have notebooks, RStudio, MLflow, scikit-learn, all of these tools that are commonly used for doing machine learning and statistical and visualization of genetics data are all incorporated into the platform. And this is all open source. So Glow is a collaboration between Regeneron Genetics Center and Databricks. We've been working together with Regeneron for the past few years. And in the fireside chat, Henry Davidge from the engineering team will be talking with Dr. Evan Maxwell about the genesis of this project and how it's evolved over time. And just in brief about Glow, to scale genomics data analysis requires teams of engineers to build reproducible, productionized genomics workflows. So Glow helps you go from science to engineering, and engineers like simple APIs, they don't want to learn the custom bioinformatics tools. They want something that's simple and reproducible or reusable. And then our other, it also requires machine learning to blend all the different data sets together. So, and to achieve this requires building on top of Spark SQL, which enables you to write code in multiple languages, Python and R and Scala. It requires you to be able to easily join in other data sources and then extend your analysis down to machine learning and data science ecosystem, and simple single line functions for standard genomics data loading and transforming. And finally, everything that we've built is backwards compatible with the existing ecosystem, the existing file formats and tool sets. But what we're enabling is the best practices in data science and data engineering using tools like Spark. We're enabling bioinformatics to be brought into that ecosystem so that engineers can build and scale these workflows. And ultimately, the goal of precision medicine requires the ability to link all this genomics data to all these other data sources. So once we're going to talk today about linking genomics with, say, clinical and claims data or electronic health record data. We've done a lot of work there, but we have other customers who work on deep learning, other customers that are working on Internet of Things devices that are streaming data in, or social analytics from real-time patient feedback, this kind of stuff. So we have customers who are doing all of these things, but the real vision of realizing precision medicine requires integrating all of these different data sources together. And that requires machine learning. So it's a monumental challenge. It's going to take years to unfold, but we're super excited about it. And we're going to talk a little bit about how this vision will unfold over the next decade. Cool. So with that, I'd like to introduce Henry Davidge. So Henry is senior software engineer at Databricks. He's been working at Databricks for the past 5 years and on genomics for the past 2 years to help build out Project Glow. And Dr. Evan Maxwell, who's a senior data engineer at the Regeneron Genetics Center. All right, over to you guys.
Okay, thank you very much, Will. And thank you everybody for tuning in today. We're really happy to be here with you. So like Will said, I'm here with Evan Maxwell, a senior manager at the Regeneron Genetics Center. And Evan, why don't we start things off by telling the audience in a little bit more detail about your background and what you're doing at Regeneron. Sure, yeah, so thanks for having me. So I work in the Genome Informatics and Data Engineering group at we call ourselves the RGC, the Regeneron Genetics Center. So my background was in computer science and then worked in genomics as part of my graduate career, and then came to the RGC after that to help work with pretty much scaling up our exome sequencing efforts. And sort of, what I focus on a lot is on the sort of the data management, data engineering side, and thinking about how to scale a lot of our analyses as our populations grow. Okay, very cool. And then to take a step back a little bit, what exactly is the Regeneron Genetics Center? Why was it started, and what's the mission, and how does it fit in with the rest of Regeneron? Sure. So Regeneron's a biopharmaceutical company, and in 2014, they founded the Regeneron Genetics Center, RGC. The goal was really to sequence DNA from populations across the world to try to catalog genetic variation and its relationship to disease. So if you think about how drugs work, is they inhibit some gene or protein that's has some therapeutic benefit. So what the goal for RGC is to build our own large internal database of human genetic variation to try to identify disease mutations in genes that would indicate whether there's some new novel drug target or validate existing programs. Because what you can do is, if you identify these naturally occurring mutations that, for example, inhibit a gene from working, and if that mutation has
a protective benefit against disease, then you can potentially design a drug to mimic the effect of that naturally occurring mutation. So that's really the goal of what RGC was founded to do, was to build this very large DNA database, if you will, paired with health records and disease information to try to identify novel drug targets and basically speed our drug development pipelines and feed new targets and genetic information into those pipelines.
So you mentioned your exome sequencing initiative there. What kind of scale are we talking about, and where is the data coming from?
Sure. So, where the data comes from. So we partner with various collaborators across the world. I don't know the exact number, but we have something about 100 collaborations to date. And most of those collaborations, we get DNA samples from study participants and some level of health information too, depending on if it's, for example, a large healthcare system or a smaller academic group. It varies a bit. But so what we do is we get those DNA samples and we do exome sequencing on them. To date, I guess, I think we hit 1 million samples in February of this year, and we've been increasing capacity quite a bit as we sign larger and larger collaborations. Our capacity right now is about 600,000 exomes per year, and that seems to grow every time someone asks me that number. Yeah, that's sort of the scale we're working with.
Wow, that's a pretty amazing amount of data. So on the data management side of things, what challenges started to arise as you got into the hundreds of thousands of exomes that you don't see if you're working with a small amount of data?
Yeah, so it's a few things. It's the size of the data and the pace at which it comes. If we're sequencing at 600,000 per year, our pipelines, we have pipelines that work directly on those single sample VCFs. For people familiar with genetic data, a variant call format is the type of file. And so there's pipelines that work with that type of data, but then we have these larger, as an example, our UK Biobank collaboration has 500,000 people. We don't just want 500,000 individual data points. We aggregate them all into what's called a project level VCF. So that's, if you think about it, it's essentially a matrix of 500,000 people and all of the genetic mutations that have been identified across that cohort. And so producing that data is sort of the first step, but we then have to aggregate it. We have to run QC on it. We have to try to use it and interoperate with other data types like the health data. So really just the size of the cohorts that we work with sort of pushes the scale boundaries of what things like a VCF and the tools built around it are really meant to handle. But there's also just what do you want to do with the data after the fact, and how do you really leverage it to get the most out of it? And for that, we essentially decided that a lot of the traditional tools that are built on flat file command line type single machine operations, just they're too cumbersome. It's a lot of work goes into these tools, but they're not really meant to interoperate together. So when we operate on a very large scale and are trying to turn things around quickly, having a much more unified and scalable infrastructure is really powerful. And so that's what led our migration to moving into Spark and representing these data sets in a more traditional, sort of not genomics focused representation.
Cool. I think that's a really great example of how we can start to use general purpose big data computing frameworks for managing the growing volume of genetic data. So as you were developing this infrastructure and resolving these challenges, how did you eventually go down the road of co-founding Project Glow with us at Databricks?
Yeah, so maybe to back up a bit, when we first started moving some of our data into Spark, there were some tools out there that we could have used, but we sort of decided to roll our own way of sort of taking our traditional data representations like VCF and make a representation that could work with Spark and Parquet files and tools like Delta. And so that turned out to be very cumbersome. If you take a flat, complex binary file like a VCF and try to represent it in this other infrastructure, there's a lot of just building complex pipelines that are suited to do exactly one thing just to get the data into this representation. So it's sort of an ETL challenge. And it doesn't maybe sound that complicated, but when you're starting to do it on very large data sets over and over again and wanting to modify little pieces, it gets quite complicated. So the idea of Glow and working with Databricks has been great with this is being able to engineer things that read directly off of, say, a BCF or a BGen or other sort of native file type representations, where you don't have to have this very complex ingestion process, that it's all sort of built into a reader. And then if you want to do something like lift over coordinates from one genome build to another, it doesn't require, updating or rebuilding the whole pipeline. It's a function that's built into Glow. So really it helps us scale and do things more quickly and more flexibly.
Got it. So you can kind of take the standard Spark view of any data set, just a normal data frame, and we extend it to work on genetic data. So you read in a VCF or a BGen file, and it looks just like you're reading an existing Parquet table or something like that.
Right. Sorry.
No, go ahead.
I was just going to say that, some of the benefits that we get out of that too are, leveraging all the things like you mentioned about data frames, all of the tools. But the other sort of less obvious things are that it allows us to harmonize our data and really treat it like a data lake. People always want to build a data lake, and we sort of are doing that with genetics data. So people and other teams can access it in very standard ways. And it's sort of a nice environment for doing that. But then it also, as we do these more high performance downstream analytics, it's also a good platform for that. So that's where a lot of our, I would say, sort of engineering efforts are pushing down the road.
Very cool. So to change directions a tiny bit, one of the exciting features that you guys have contributed to Project Glow is GlowGR, a new method for whole genome regression on top of Spark. Maybe you could talk a little bit about what led to that work and what sets it apart from other tools.
Sure. Yeah. So in addition to the genetic data being large and hard to manage, there's a lot we have to do downstream with it. The most obvious thing is correlate it with disease phenotype information. So for people who are familiar with sort of the statistical genetics field, there's what we call genome-wide association studies, where you take every disease mutation that you've identified in a population and you compare it to every disease that's represented in that population as well. And so if you have millions of genetic variants and maybe thousands of phenotypes, you're doing an all pairs comparison of that. And so it's a pretty heavy duty compute task, and it's a very brute force thing to do. One thing that's sort of occurred in the field over time is what are called linear mixed models. And there's sort of a specific flavor of what a linear mixed model is in the genetics context. And there's a lot of purpose-built tools for doing it. And effectively, all you're trying to do is capture some effect of things like relatedness in your population when you're doing these genome-wide association studies. And so the tools that are built for that, it's a very active area of research. They're very heavy-duty compute just to get this sort of whole genome part of that method built. And so really, it's a question of scalability. So we could, as we do thousands and thousands of phenotypes on hundreds of thousands of people, the compute gets quite big. So, anything that can make those analyses faster is, has cost savings, but it also has time savings, and it makes it easier to iterate on analyses as we decide you want to modify some model that comes out of this. And so that's sort of, I think, what the primary driver was how do we make a more efficient engine for this? But also, right, all of the other features of that I mentioned with Spark being, we sort of treat data more like a database, and we can standardize these sort of downstream analytics and representations to be more interoperable. And so, so that's sort of what the main driver was. But what we didn't want to do was just take these tools and figure out how to parallelize them with Spark in a single machine approach. What we really wanted was to sort of re-engineer it to be a distributed computing problem. And so that's what we're proud of with GlowGR is it was the first method to do this whole genome regression in a truly distributed fashion. So it will help us as we scale towards more samples over time.
That's very cool. So in addition to Glow, were there any new features in Spark that made it easy to write this kind of complex model?
Yeah, so as you know, Databricks has put a lot of effort into building out the Python and Pandas UDF interoperability. So some of the previous work we had done in this space, we tried to develop a lot of these tools using Scala, native Scala. And as people who have written a lot of linear algebra know, Scala doesn't have a very robust linear algebra packages. So being able to develop it entirely in Python and using the Pandas UDF operations with PyArrow have made it extremely fast and a lot easier to develop in general. And I think it's a lot more readable as well. So we're hoping that we get more contribution from other people in the community.
Awesome. Yeah, it's really great to be able to put out open source work that people can understand and hopefully modify themselves. So it sounds like a pretty powerful concept under GlowGR that basically we can look at a cohort of genetic data and we can kind of look at all of the variants a person might have and give some kind of phenotypic prediction for them based on all the variation we see. I think you talked a little bit about GWAS here. Do you see any applications of this work to other fields or applications in genetics?
So I guess you're alluding to things like polygenic risk scores, or do you mean sort of outside of genetics entirely?
I was mostly thinking of polygenic risk scores or something else in genetics that I don't know about. If there's something outside of genetics, feel free to mention that too.
Well, yeah, I mean, to the latter point, what we did is a fairly basic way of doing a linear mixed model, and I'm sure it would be applicable to other fields as well. And it scales quite well. So, I think there's potentially ways to sort of migrate that into other domains as well. But to the genetics point, if you think about what a whole genome regression is for this linear mixed model concept, is very similar to what's often called a polygenic risk score. So the idea is, can you predict somebody's sort of risk of having a particular disease based on their entire genome? And this is sort of different from a GWAS, the genome-wide association study, because you're not testing one variant against one mutation at a time. You're sort of treating it as more of a predictive model. So as the methods get more powerful, you can predict that more and more accurately. And so that's one area that we're sort of most immediately pushing, is figuring out how to sort of modify the method or make it more extensions to it towards the polygenic risk score domain.
Cool. And then as you get these results back from your association studies or your polygenic risk score analyses, how do you go about making that data available to other people in the organization?
Yeah, so we have these data representations of the raw, what we call the sample level data, right? The genetic data and the phenotypic data for every patient or sample that we have. But then there's the output of these association studies too. And just like, if you compare every mutation to every disease, that gives you hundreds of millions, billions of summary statistics to work with. And so we also build those into delta tables as well. That gives us essentially a massive database of genotype, phenotype associations and some statistics around them. And we build different downstream methods and analytics that operate off of that data representation as well.
Cool. And do those downstream applications read directly from the delta table, or do you move the association results to some other system?
Yeah, so we do read directly off of the delta tables. We have various ways we do it, but it's we essentially build a database in Spark and Delta based on these association outputs.
Cool. Yeah, I can imagine it. It seems like it's a lot easier to, this is a major advantage of using kind of general purpose computing frameworks and programming languages, is that we can kind of use a small number of systems to go from thorough ingest to the statistical analyses to making data available to a broad base of people who might want to be able to consume it.
Yeah, absolutely.
So you talked a little bit about how, in addition to having a lot of genetic data, you have tons of medical records and other sorts of health data that you want to integrate. Is there any particular challenges that came up with having so many disparate sorts of data?
Yeah, there's a lot of challenges. I think, I mentioned that I work in genome informatics data engineering. So at RGC, we also have clinical informatics groups. And I consider ourselves lucky a bit because DNA has a very standard, we compare everything to the reference genome. It's very standard. But when you deal with medical records, it's much messier, and it's also less standardized from collaborator to collaborator. So there's very complex and time-consuming processes to try to standardize clinical data to the same degree that we can standardize genomic data. But I think really the goal is we sort of have a target for this is what we want the data to look like, right? And it's something very similar to what we do with the genetic data in Spark. We want to build databases and tables that store the data in its standardized format, and then that lets it be it's much easier to interoperate with, aggregating the genotype and the phenotype information, but also pulling in, aggregating data sets between collaborations where, one might be a health record data set, another might be an academic data set. So trying to get them sort of on the same page, if you will. And I think, yeah, that's a big challenge. We also do things, try to pull in external data sets. So sort of your standard public genome genetics resources that, let's say you want to pull in annotations from, EXAC or some of these other large consortia. We use the same processes to ingest sort of these external annotations and make them look like databases as well. So that in the end, it's just a large data lake of databases from the genetic data, the clinical data, and external resources.
It's cool to see how the same solutions keep popping up across domains. It seems like it's really useful to you guys internally to be able to kind of represent all of your data in the same way and have these unified sorts of data lakes. So then looking forward a little bit, what are some things that you're excited about adding to Project Glow, I guess adding to Glow that you think are going to kind of make a difference and help other people scale to the sorts of data volumes that you work with at Regeneron? Sure,
yeah. I think sort of our immediate interests lie on sort of the downstream analytics pieces, trying to do more with Glow GR and build some other types of models, maybe the polygenic risk score direction. I think there's a lot to be, done still on the raw, sort of the ingestion of the genotype data itself, the things like the VCF readers and the things like liftover and normalization functions. But I think sort of our immediate interest is in trying to build these more high performance analytics that work with Spark. And I think Glow GR is really sort of the tip of the iceberg for that. So we have a lot of interest in moving in that direction.
Cool. Watch this space, everybody. So to wrap up, I think we have about 2 more minutes. What are you excited about seeing in the RGC in the next few years?
So as I mentioned, our sequencing capacity increases all the time. But so do our collaborations, right? The sort of the size of the collaborations. And so the data sets we're building get bigger and bigger. So I think, the scale question will always be something we're trying to address and sort of be a couple years ahead of. I would say we also have plans to do some whole genome data. So basically it'll increase the dimension of the variant dimension of a matrix. So that's sort of another thing that's going to be pushing scale a bit more. We also are working to, as most of the field is, try to get genetic data sets that represent more diverse populations for people sort of in the field. That most public genetic data sets sort of across the world are very heavily sort of European ancestry, and that's just not, the best way to be looking at human variation. So we're trying to get more diverse populations and larger data sets at the same time.
Okay, very cool. All right, Evan, thank you so much for joining our webinar today. It's been an awesome chat.
Sure. Yeah, great. Thanks for having me.
Cool. So I think we have about a 10-minute break now.
Yeah, and during the break, feel free to, if you don't already have the Databricks Community Edition or a Databricks account, to sign up. It's free to sign up for Databricks Community Edition. Just go to databricks.com/try-Databricks. And then throughout the workshops, we're providing notebooks that are available online that you can import into your Databricks Community Edition. We'd recommend just listening along throughout the workshops, but then after the workshop, you can go ahead and try it for yourself if you're interested. But we're going to break now, and we'll get back started at 10 a.m. Pacific, 1 p.m. Eastern. Thanks, everyone. Okay, we're going to get started in just a minute here. So just to recap the agenda for this morning, or this afternoon, or this evening, wherever in the world you are. We're going to have a workshop next where Karen Feng, who is a software engineer at Databricks, who's done some really incredible work on accelerating variant analysis by using Spark to distribute some of the GATK's algorithms for combining variant call format data on a really massive scale. And then after that, Amir is going to go through how to do statistical genetic analysis on genetic variant data with GLOW. We'll have another break at 11:15 Pacific. And then we're going to end with Kievash, who's going to talk about how to do ultra scalable whole genome regression with GLOW GR, which is a really just incredible innovation that was developed with Regeneron and Genetic Center and Databricks. So I will pass over to Karen for the first workshop. Over to you, Karen.
I think I'll have to take over screen here.
Okay, I'll stop sharing my screen.
Here we go. Yeah. All right. Thanks for joining, everyone. Today, I'll be discussing our pipelines, but in particular, joint genotyping on Databricks. This leverages Spark and Delta Lake as well as GLOW. So just to get a 1,000-foot view of the problem, we want to do aggregate analysis on genetic data. This is kind of a given at this point. With the amount of information that we have, the more data that we can get, the more information that we can aggregate. On the other hand, the more data that we have, the harder the problem. So for example, we had a customer who wanted to generate these statistics using Plink, which is a single node tool. It's been used historically on the scales that are commonly known in the genomic space. But as we get thousands of genomes, this process gets longer and longer. And if you're making this report once a month and this report takes a week to generate, you can imagine that we simply run out of time. So how do we work with this large scale where we have so many disparate files? Joint genotyping is kind of the answer to this question of how to deal with aggregate analysis. At the core of joint genotyping, we are taking a bunch of small flat files called GVCFs or genotype VCF files, and we are squaring off the calls across all of the samples at all of the sites. And this allows us to create a single source of truth. Each of the rows in the GVCFs represents a block of the genome for which a single sample may or may not have a variant from the reference genome. And from joint genotyping, we would create a single project level VCF which contains all the samples, and each row represents a site for which at least one sample we've determined has a variant call. And in addition to creating a squared off matrix where it's easy to perform aggregate analysis, we can actually see these statistical improvements when it comes to finding real signal. Here I'm zooming into a single site on chromosome 20, position 18 million or so. And we have a single sample up top. This is from a VCF file. You're probably quite familiar with this if you're working in the genomic space. And you'll see that for the sample, we believe that we have one reference allele and one alternate allele, noted by GT01, and the quality score is less than 200. However, if we include the data from two other samples, HG00268 and NA19625, we're able to improve the quality score at this site significantly. And in essence, what we're seeing here is improved confidence in our variance calls based on evidence across an entire population. Here we only have 3 samples, so it's kind of a toy example. We can see that even in this setting, we can see improvements. And this confidence is reflected in improved accuracy across the entire genome, not just for this single site that I've cherry picked. So if you remember your high school statistics, recall is the percentage of positives that you were able to identify in your analysis. And here with joint genotyping, by including 2 other samples, you can recall significantly more of the variance. Here, indels are insertions and deletions, and single nucleotide polymorphisms are SNPs. Unfortunately, although joint genotyping is useful for aggregate analysis and kind of forms the first building block on which you can perform your statistics, it is limited historically by the fact that the tools are built for the single node. And so we had one customer who wanted to run joint genotyping on 5,000 genomes, which isn't actually even too much nowadays, but it took 6 weeks. And this is part of a larger problem within this space. These tools that were built historically are meant to be easy to use. They're built via the command line, they work out of the box, but as you start adding more and more samples, you start running into limitations. This could be in memory, on disk, and just pure runtime. But the tools that were actually designed to scale along these dimensions can be quite difficult to use and set up. And I'll go into a little more detail to present examples on these specific problems. So here I've pulled out the command line tool from the Genome Analysis Toolkit out of the Broad on how they recommend running joint genotyping if you have a small number of samples. And there's a reason we've limited this to 1,000 samples kind of as a maximum. You'll notice here that we're working with flat files that have to fit locally. And the steps are fairly straightforward. You have combined GBCFs, which takes a bunch of these single sample GBCFs and makes one big multi-sample GBCF. From this, we can perform genotype GBCFs, which actually runs the joint genotyping algorithm. Unfortunately, combined GBCFs, the first stage of this process, is known to not scale well for the 1,000+ sample size based on the underlying algorithm, and actually has an exponential runtime. Unfortunately, the tool that was meant to address the issue here with combined GBCFs not scaling well is quite difficult to use. Here, I've outlined the architecture of Genomics DB, which tries to solve this issue. You'll see here that there's a tile DB array storage manager on the bottom, a variant storage manager on top using C++, and then 2 separate stacks on top of that. And the issue that's being reflected here is, I think, a common trend in genomic tool development. Rather than consider genomics under this wider umbrella of data science and data engineering, a lot of these researchers end up rolling their own ad hoc tools, which creates an unnecessary headache, given that such tools are designed for scale if we actually use the common standard ones. And in addition, these tools are simple to use for a general use case downstream, such as for, as we'll describe later, doing basic statistics and GLOW WGR and so on. So as I already spoiled in the title, the tools that I'm discussing today as part of the standard toolkit for data scientists and data engineers are Spark and Delta. If you're here, you probably already know what they are, but just to refresh you on them, Spark is a big data processing engine which scales well for this sort of aggregate level analysis. It's easy to use along a variety of dimensions, but here I'm highlighting the fact that Spark is built for people from different backgrounds. Bioinformaticians, for example, often want to use SQL, Python, and R, while as a computer scientist myself, I often want to write Spark code in Java and Scala. And so I can write my code in Scala and expose it to the end user who wants to use Python downstream. And the other flip side of this coin, we have the processing engine. We also have a file format which is compatible with Spark called Delta. And this is designed for scale as well. Delta Lakes can handle petabyte scale tables and support version updates, which is reflected in the fact that genomic data often comes in batches. So you'll often want to perform appends. All right, so GLOW is really designed to work as the in-between between these common tools within the big data ecosystem, here Delta and Spark, as well as the tools in the genomic space which have existed and have these tried and true algorithms which we would like to use, such as the GATK. In particular, GLOW can be used to convert between formats or Java classes that are compatible with the HDS JDK, which underlines the GATK, and data frame rows from Spark, which can then be written out to Delta Lakes, or to VCFs using GLOW. And this allows us to leverage these existing easy to use genomic tools with other existing scalable big data tools rather than rolling our own ecosystem. So here I've zoomed in quite a bit, and here I'm just describing the architecture for our joint genotyping pipeline as part of our unified analytics platform for genomics. At the first step, we have a series of flat single sample GVCFs. These are specified in a manifest. I'll describe this in the demo in a little bit. But the idea is that we take all of these and we ingest them using GLOW into one big data frame. And this data frame unions the single sample variant calls across all of the samples. GLOW is also compatible with other data sources such as BGens, but here we're just describing the VCF file format. We then use Spark to partition the data. Here we're creating essentially bins across the genome. And this is because the downstream algorithm that we want to use, the GATK genotype GVCS algorithm, expects blocks of data that come in with one GVCF row per sample in order. And so here we're partitioning the rows for these blocks and then sorting them within each of the blocks. Each of these partitions are processed in memory, as is done in Spark, in parallel, of course. And using genotype GVCFs, we process each of these sets of single sample GVCF rows at a time. And the algorithm will choose whether to output a PVCF row if it believes that there's a legitimate variant at that site. And this can then be output to a Delta Lake at the end by default, although we can also output to a project level VCF or PVCF, which can be sharded or as one big file if you want to share with your collaborators. Although the Delta Lake will give you the biggest benefits when it comes to querying and downstream analysis and so on. Joint genotyping is designed in this architecture to be inherently scalable. And here I'm just showing some of the results that we've had on fairly small data sets. We've run on much larger sample sizes. But here, as we scale up to say 500 samples, we can say that adding more samples increases the runtime within a reasonable scale. You'll see, however, that there is a bit of a bump at the end. That's due to spot terminations. Here, I was being cheap. Spot instances can be used in AWS and Azure, and they can reduce your cost significantly. And all you're really saying is to the cloud provider, you can take away my instance, and in return, you'll allow me to pay a little bit less for it. Spark is able to recover from these instances being taken back by the cloud provider. Here, I was probably in a very popular instance, and there was some contention. But here we have fault tolerance so that we don't have to deal with the problem manually. And on the right side, I have a graph that demonstrates that, if I'm feeling impatient and I just want to get my process done as quickly as possible, no matter the number of samples, we can just throw more workers at the problem to reduce the runtime. And although I showed an architecture that I think is quite straightforward, within Databricks, you don't actually have to think about all of the pieces of the puzzle when it comes to GLOW and Spark and Delta and how everything fits together with the GATK. We abstract away the boilerplate in this pipeline by saying, all you need to tell me is what are your input GVCFs? How do you want it to be output? We'll put in a Delta Lake by default. We can also put in a PVCF. And you can also tell us how fast you'd like the pipeline to run by saying how many machines you're willing to throw at the problem with the cluster configuration. All right, so that's essentially most of my slides. We're going to go ahead and start on the pipeline demonstration now. And if you follow this link, you'll be taken to the demo notebook, which you'll then copy into your community edition workspace. But I'll walk through this step by step. So if you go to our documentation, you'll see the joint genotyping pipeline. It describes in some detail what I went over today, including additional optional step, ingesting variants into a Delta Lake. And this here kind of serves as a proxy for what would happen usually in combined GBCS or with Genomics DB. By default, we skip over an intermediary Delta Lake, which contains all of our single sample GBCF rows, but you can put it in there if you believe that you're going to have to process the data multiple times, or if you're going to have more batches coming in and don't want to re-ingest your data. Although we're always going to perform the joint calling, of course. And in the setup here, we're going to go over shortly how we create a cluster, although we do already have some of these recommendations when it comes to worker numbers and sizes that we believe work well at different scales. We'll also be going over some of the parameters that you can set. I mentioned earlier that you'll have a manifest, which contains all of your input GBCF files, and you'll just set where you want the data to be output and how you want it to be output, in the sense that if we already had a Delta Lake, whether we want to append to it at the intermediary stage. We can also export to VCF and perform filtering for targeted regions as required. And as I mentioned earlier, here we have Delta files and VCF files if configured as so. And one of the key steps here is that you have to set the reference genome. We have already set the environment variables for ref genome ID for GRCH 37 and 38, so that it pre-downloads the reference genome onto your cluster on all of your worker nodes, so that you don't have to do it step by step. But if you want to use a custom reference genome, we have instructions for that as well. The manifest format simply looks like a list, nothing that exciting to it. You can put blobs, however, and each of the rows can also be a directory rather than just a flat file. All right, and if you follow the link, you would have been taken to this notebook here, which we'll be importing into our workspace. But the first step in Community Edition is to enable the genomics runtime. You'll see this in the admin console up here. And all this means is that it allows you to enable the genomics runtime in addition to the default runtimes, which include machine learning and our vanilla runtime. The genomics runtime includes Glow, as well as, I believe, SAM tools and these common genomics libraries, so that you don't have to do everything from scratch every time you want to create a cluster. And here I've already created a cluster that we can use just for the base level investigation of what the pipeline is doing. And I can go ahead and edit and just show you how you would create a cluster with this sort of setup. You can set the cluster name here. And in the Databricks runtime, you would choose which runtime you would like. I'm on an internal cluster, so there's a lot of these more custom versions, but you would choose one of the genomics runtimes. LTS means long-term support. And here, these are recommendations that we'll be using for the job later as well. We're going to enable auto scaling, which allows the cluster to have more or fewer workers based on demand, which allows you to reduce costs and scale up if we have more if we want to throw more workers at the problem without doing it manually. We also have auto scaling local storage so that you don't run out of disk space. And here on an interactive cluster, we also have the setting to terminate after so many minutes of activity. By default, I think it's 45 minutes. I just increased this for this specific demonstration. And when we enable auto scaling, you'll see here that you can configure the minimum number of workers you would like to have on a cluster, as well as the maximum number. And the recommendation for joint genotyping is that you use i3.4x larges or larger, or the parallel on Azure. And we're using the I instances because they're storage optimized. And for this specific cluster, we've set an instance profile so that we can have the right permissions if we need to write to a bucket. We can set an environment variable here for a reference genome. We don't have to right now because we're just doing exploration, but this is where you would say, I want to work with reference genome 37 and so on. And finally, here is where you would set an init script. If, say, you wanted to download a non-standard genomics library and have it installed on every worker or node in the cluster generally, you would create an init script that downloads it and then puts it into a specific machine, which would then be distributed across your entire cluster. All right, so this cluster is already spun up, so I'm not going to mess around with it any more than I have to. And the first step in creating a pipeline that works for your specific data set is to make a manifest. Here, I'm assuming all of your data is already in an S3 bucket or an ABFS bucket. Although in this specific demonstration, I'll be going over the public data sets that we've distributed via the DBFS or the Databricks file system. And here we're using the Databricks utility ls, which is similar to what you'd be familiar with, say, with bash commands. And we're just checking out what's happening within this directory. It looks like we have two BG zipped GVCFs, a README, as well as a manifest. Well, let's take a look at what the README has. Here we're using the DBFS fuse mount to access this README as if it were on my local machine. And you'll see that the files that we've created here are from the 1000 Genomes project, filtered to only contain records for chromosome 22, just so we can run this demonstration a little bit faster. And within the manifest, we can check the contents by using Spark, although this is probably overkill, it's only 2 rows. But here we're using the CSV reader to load it in. And we'll see that, okay, yeah, each row is one of the GVCFs within the same directory. So this is just relative to where the manifest lives. Let's also take a look at what the GVCFs actually look like. We're going to use the Glow VCF reader to take a peek. Here, you'll see that we'll just set the format to VCF, and everything else is hidden under the hood. We also have a fast VCF reader, which is coming in soon, so keep an eye on that. But what this looks like is actually quite similar invariant of which file format you end up using. Here we're using VCFs, but if you read in data from Plink files or BGen files, they also look quite similar. By default, we're always going to have specific columns, such as the contig name, here chromosome 22, the start position, which is actually the VCF start position -1 because of the way the VCF spec is set up versus the way we configure our variants. We have an end position, which is actually much farther than the start position plus the size of the reference allele, which is what it would be by default. Because we're reading it in a GVCF, we have blocks, which can extend for quite a few alleles. So you'll see that this specific one is about 800 base pairs or so. And in this column, we're going to have the exciting parts. We can demonstrate here that this is a single sample GVCF. The sample ID is HG0096. And we have our Fred likelihoods, which are used later during the actual joint genotyping algorithm to determine our calls. And you'll notice here that we don't have any calls because we're not sure quite yet what it actually will look like. All right, now we need to actually make a manifest. It already exists in the Databricks data sets, but here we're just going to make it again just so that you can see what it looks like. We're using the DB utils functionality here, and we're going to write the manifest a little bit differently this time. Rather than pointing to the relative VCF path directly, we're going to use the glob pattern so that we're saying any file under DBFS, Databricks datasets, HG0096, and maybe anything after that, that's going to be representative of our GVCF rows. And so once we put it in this path, here I'm using DBFS root under my own name. You'll see that the rows look like this. All right, so once the manifest is in, we need to make sure that we have our other parameters ready for our pipeline. So the pipeline is actually fairly straightforward. There's a bit of boilerplate, and let me just break it down for you, each of the different parts. The first step is we actually create the pipeline. This contains all of the architecture that I described earlier under the hood. But we will reveal to you what the parameters you can set are. And this data frame will demonstrate in a little more detail what each of them are. So here, export VCF, fairly straightforward. You can also specify if the VCF should be sharded or as a single file, and so on. This specific notebook cell tells the notebook to create these widgets up top, which you can set manually if you're running the job interactively. We'll be running it as a proper job, which will reduce costs because job clusters are a bit cheaper. This cell just reads in the parameters from the widgets, which we will then pass into the pipeline and actually execute. And this final one here simply goes ahead and tells us what's in the output directory. So we can make sure that it contains what we expect. We can perform our querying and so on. Okay, to actually create a cluster, you would go over to the jobs page. So that's on the left side here. And you would create a job. I'm going to skip over to one that I've actually made for the joint genotyping demonstration. Here, I've imported the notebook from our public documentation. How you would do that is to go over to the Databricks link that we had earlier. You're just going to copy this. Go over to your workspace and import. And you can go ahead and just import from URL here. I've already done this here, so I'm not going to redo it. But once we've imported it, we will set the jobs notebook to what we actually want to run on our cluster. Then we can set our parameters. We can edit it with the key values or with the JSON if you're more comfortable with that. Here, I've already configured, I want to see a VCF at the end, but I didn't set export VCF as single file because I'm okay with it being sharded. I've also set the manifest that we created earlier with the glob input files, as well as my output directory. And I've set the replay mode to overwrite because I ran this job a couple of times, and I just want to make sure that every time I run it, I rewrite my output. Here, I configure my cluster based on the guidelines from the documentation. I was going to skip back to that to show you what that looks like. Here you'll see a JSON, which specifies we want to use auto scaling. I'm willing to go up to 25 workers, etc. And so you can just go ahead and just copy this. If I can get this working. Give me a second. There we go. Okay, I'm going to copy this. And when we head over to our cluster, we can actually edit it as a JSON. So I essentially just copy pasted this here, so that you don't have to do any more fuss than you have to. And in that JSON, we also included the reference genome is set to GRCH38. And once all of that is ready and you're feeling ready to go, you can configure a few more things if you'd like. For example, you can set alerts to let you know when your job is starting, when it's succeeded, or if it's failed, as well as some other advanced settings. And I just went ahead and clicked Run Now. And after it's done, this took 9 minutes or so. A lot of it was just set up. Here, we're just running on 2 samples, so it's quite quick. But we could have used far more samples. We could have changed the worker size. There are so many things that we could have configured to do this demonstration a little bit differently. But this was just the easiest one out of the box. And if you want to take a look at what the different Spark metrics are, you can also click through. This will show you the Ganglia historical metric snapshots, or the Spark logs, the Spark UI, etc. We're just going to click through here. And this will show me how my demo went. It looks like everything ran through successfully so far. The parameters were set based on my job parameters. We use the manifest. The VCF compression codec is default set to BGZF in my output. And the job itself actually ended up only taking 2.5 minutes, once we exclude the cluster setup and configuration. And in the output, we'll see a delta table under output genotypes, as well as a BG-zipped VCF here, which is quite big. And to actually look at the results, I made another notebook just to peek about. And we'll see here, as we saw earlier, we have these two tables. The first one is our delta table. Here, using display, we only show up to about 1,000 rows, just because you don't actually need all of it if you're just looking at it interactively. Although, of course, you can get all of them if you needed to. This just makes it a bit faster. And you'll see for chromosome 22 at this specific site, we scroll all the way over, and there are 2 samples under here for HG0096 and 97. And the calls here represent that we have 2 samples for which both of them had only alternate alleles. And all of these sites are variant call sites. They aren't GPCF blocks because of PVCF, so it'll be quite a bit smaller than all of our input files. And we can also read this in from the flat VCF file that we output. It just takes a little bit longer because we're loading in from a flat file, but it might be easier, say, for your collaborators. All right, that's actually all that I have for the pipeline demonstration. But to zoom back out to what Databricks has outside of joint genotyping, we have pipelines that are built for DNA sequencing, RNA sequencing, tumor normal sequencing, as well as variant annotation pipelines for SNPF and VEP. All of these pipelines look quite similar. I showed you a notebook which has all this boilerplate in it to create widgets, to read in parameters and so on. And all of them basically just look like that on top. Under the hood, however, our architecture is based on the same principles, which makes them straightforward to set up and scalable to your use case. If you were at Spark Summit a 1.5 years ago, I was talking about how we had just made this joint genotyping pipeline, and I was excited about the architecture of it. But now we're talking about it because we've seen it work successfully for a lot of our customers, and they just seem to work right out of the box. We have a customer who ran through 50,000 exomes for 7,000 DBUs, which is this Databricks pricing unit, but that's less than $500 with fairly minimal introduction. And so we look forward to seeing more users on the pipeline. We recommend this pipeline for users at this sort of scale with thousands of genomes and tens of thousands of exomes. That's about all I have today, and we look forward to talking to you more about your use case. If you have any questions about our pipelines, especially joint genotyping, feel free to drop in the Q&A. After this workshop, we're going to be going on to Amir, who will be discussing how to characterize genetic variants with GLOW. So with that, I'm going to go ahead and call Amir up. He's one of our senior solutions architects on the healthcare and life sciences vertical, working on genomics in particular. And he'll be able to walk through some of the really cool base functionality in GLOW and open source. All right.
Okay, thank you very much, Karen. As Karen mentioned, I'm a solutions architect here at Databricks. I have been working at Databricks for the past over 2 years now. But my background actually is in statistical genetics, and prior to Databricks, I worked as a staff scientist at Ancestry DNA. And at the time that I worked at Ancestry DNA, it's a long time ago, we just surpassed they surpassed their 1 million customers that they had in their database. And this was array data. Based on the latest public results, we know that they have more than 15 million. The same thing with these commercial companies such as 23andMe. And more, not only they have array data, they are doing whole genome sequencing, whole exome sequencing. And one of the reasons that I joined Databricks is that in between my Ancestry DNA and Databricks, also I worked as a senior data scientist at a large e-commerce company. And that was the first time that actually I was introduced to Spark. What was really interesting to me was the power of Spark and parallel computing, that it is already being used in traditional FAANG companies, that you can process petabytes of data, whether it is stream or batch, and do all your data manipulation at scale. And this reminded me of when I was trying to do some preliminary GWAS on the million array data we had. And Plink was not really sufficient. Some single node R code that I was writing wouldn't really scale that much. And that is where it clicked, that really we need to bring the power of Spark to genomics field. And this is I'm super excited about this opportunity to be here and also to work with a lot of customers that are actually using the power of Spark, in particular Project Glow, to advance genomics initiatives. One of the things that we know in the field in genomics, when you're looking at the genomics data, by itself, genomics data is, it's really good for a lot of, if you are doing something evolutionary biology studies, you want to look at selective sweeps and interesting signatures, across. So there's a lot of information for sure. But really, when it comes to getting some business value out of it, the well-proven use case for genomics now is, as Evan from RDC mentioned, is running GWAS and getting data, informing your drug target discovery based on the hits that you get. And as we know, by itself, you need to get that phenotype data. And more phenotype data is coming in a variety of ways. UK Biobank data is a great example that you're scraping phenotype data from electronic health records, you're scraping data from even images, and you want to have a unified platform that to be able to combine all of these data sets and use the same APIs that you're using for analyzing your other data sets. You can use the same APIs to use for genomics data. And this is, as mentioned, William also talked about it, Karen mentioned that. I really want to emphasize that this is where we see really the power of Glow library that is bringing standard data science toolkits that are used across the board to the genomics field. When it comes to Databricks specifically, from an architectural perspective, because your genomics data is being treated as any other data frame in Spark, you can leverage all the existing tools. For example, we have the Delta Lake. So Delta Lake is a data lake with superpowers of data versioning and also high performance in a way that you actually, specifically for a lot of R&D, you can treat that as a data warehouse in a way, in the sense that you can have unstructured data in a data lake, but at the same time, you have schema enforcement, evolution. So they have a lot of governance on your data that you need. And most importantly, that what really matters to scientists is reproducibility down to the data level that you have. So you have a data versioning that you can even query specifically from a specific version of your data. And also, when you are working within this architecture, you can also tap into the wealth of other tools that we have. For example, within the use the machine learning workloads. More and more we are seeing, especially in the RNA field, that some deep learning workloads are creeping into genomics. Obviously, a lot of it's more advanced, different ways of doing statistical analysis. That's in my books. Deep learning or machine learning as a whole is statistical learning, and you can use that. All of these libraries within the same environment, and also all of this is managed through MLflow. So MLflow is the way I describe MLflow, despite the ML can be misleading, but think about it as this is a digital lab notebook. So bench scientists that I work with always, we had really good ways, they had really good ways of recording every experimentation. And then you can go back and look at the notebook to, for reproducibility, or that goes in your, methods section of your paper. We didn't have, in a dry lab practitioner, statistical geneticist, personally, I can say I wasn't quite, rigorous with keeping track of what I do. And I'm really happy when I see MLflow, is that MLflow is this tool that you can track all experimentation. Every run of your notebook actually is tracked automatically, especially when you're running on Databricks. If you have any metrics for your experimentation, you can track that. And then you can search easily through all of your experiments and come up, which run of this experiment had the highest, I'm just give an example, concordance result between this variant calling and GL, for example. And there's a lot of other tools that I'm not going through that, but I just want that to be, also keep that in mind, that this integration within the broader ecosystem is where we are seeing a lot of customers, especially in the pharma R&D, are seeing the value, getting the value out of it. One of the, William brought this graph up. This is in terms of this architecture that we have for recommended architecture for Delta. You can think about Delta as, we have this kind of data refinery that you ingest initially, you ingest your data set into this bronze table of your Delta lake. And then you perform quality control, depending on your use case. And then QC results are being written in the secondary layer, which is this, we call it silver. And then next level, you can write your results into the gold tables. Canonical example is a GWAS. You have an initial data set, UK Biobank, for example, or FinGen data. You ingest that as is. Next level, you want to do some QC control. So QC can happen in both on a variant level or a sample level QC. Most of the QC operations actually involve basic summary statistics calculation that I will cover here. And this is the value that you're getting when you're using this library, is that all of this is streamlined. It is done in a distributed fashion that you can scale really well. And then, results, you see the results of the QC variants, you can see it on the silver data, where next level is that you're doing, GWAS, or as Kiawesh will be talking about, when you're doing a whole genome regression analysis. So within this architecture, so this is something to keep in mind, that you can use this architecture to really organize your data and organize your pipelines. So the value added is that you're not going to repeat experiments, and if you're on any team, you can have access to that directly. Maybe it was also mentioned, one of the features, like, since we are working on a Databricks notebook environment, is also you have all of revision history of any experiment, any notebook, which you can also link with GitHub. I mentioned about experimentation, and also you have to collaborate in features of commenting. Again, this is playing into the whole scalability of your workloads. You're scaling both on the machine size flow and also on people, collaboration. What really we want to do is that, I give an example myself. Again, in my past job, we had teams that they were working on records and genealogy, the core ancestry, the massive genealogical data. And we had a lot of teams that were using data science toolkits, and the computational biology teams, mostly we were using our own VCF tools, Plinks, etc. So having one platform that actually now collaboration can happen across teams, I think it's super valuable, especially if you're working in those kind of environments. On the Glow side, Karen showed how you can easily, as an running example here that I'm running, is that you start with existing formats. VCF is a common format, BGen or Plink file, doesn't matter. And with one line of code, you're ingesting that in Spark data frame. So what I'm doing actually, when you're ingesting this, as you see immediately, there is a structure given to it. So you're going from a semi-structured flat file, which is VCF or Plink, to a structured data, which is, in this case, is a Spark data frame, that you can view it in a tabular form. It has all the information here, including all the info fields that you get from the the header of your VCF file, plus genotype variants. But the interesting thing is that, and specifically when you're working on the platform, now I can treat this as any other table that I can write a SQL query against it. Let's say, maybe it's not the most meaningful query, but let's say I want to see how many variants I have that the reference allele is A, but I can just write it as a common SQL that most data analysts are familiar with, and run that. And keep in mind, SQL is one of the oldest languages, and by design, it is designed that it is very close to natural language. So it's really easy to pick up. So the value is that on the other side, we have statistical geneticists or computational biologists. We are working with the data from as a data scientist perspective. We are comfortable writing code in Python or etc. But maybe sometimes the results of that value, some clinicians or clinical researchers from more of the bench side of the research might be interested in something. And I would say, I have encountered a lot of people that they don't know Python or R, but SQL is very easy. So this is really powerful in the sense that we can all work together regardless of the level of familiarity we have with coding. So on this topic, let's just go and see how can we do some kind of simple statistical genetics. And when I'm saying statistical genetics, it goes from very basic statistics of, for example, looking at allele frequencies or homozygosity, or runs of homozygosity, etc., on a sample level, all the way to GWAS, and which Kiarash will talk about. I would say it's, especially when you're looking at a GLOW-GR, is one of the most cutting-edge statistical genetics workloads. And you can do all of that within this framework. As an example, let's say I want to look at my data. I have ingested the data set, and I want to look in basic sample level statistics. So what for example, I want to look at, I want to look at the call ratio, because if you have, especially for sequencing, and you want to exclude samples that they don't have good calls, or the number of heterozygosity. And the heterozygosity actually is a really good, if you have higher outliers in heterozygosity, that is an indicator of sample contamination, for example, and especially in the when you're collecting massive on a commercial side. So these are the basic QCs that you can apply. But with a simple SQL-like query, you can get all of these results, and within the platform, you can also visualize the data. So in this case, for example, if you want to look at a correlation between heterozygosity and deletions, you can just quickly visualize that within this platform. As you will see, this is not the only way for data visualization. You can use a matplotlib or ggplot in R. As a matter of fact, if you look at the documentation for GLOW-GR, the sample quality control, you have a lot of built-in functionality within that. However, what is really important is that also you can do your own user-defined matrix. So there are two ways of doing this. You can use this, the method aggregate by index, or if you're more on the Spark side of things and you're comfortable with Spark, you can actually use Spark native, explode function, just flattening, creating this really long skinny tables, for example, flattening your schema and doing any analysis that you want, and then collect it, aggregate it and take it back. Just one aside, any analysis that you're doing on your data, it boils down to two basic functions: a grouping of the data set, you want to get some samples on a grouping, and then applying an aggregator or a function that calculates the summary statistics. So the advantage here is that on genomics data, you can actually now tap into wealth of Spark MLs. If you want to do, for example, PCA or anything like that, you can use Spark ML functionality, or use straight up R functionality for your statistical analysis here. So this is on the sample level. Let's see if I want to also look at, on a variant levels, statistics, summary stats. So, classic example is that now I'm looking, for example, I want to look at the allele frequencies, or as you'll see, or Hardy-Weinberg. I want to correct for Hardy-Weinberg disequilibrium. This is within the Glow library. You have again built-in functions, for example, call summary stats. By default, it generates these columns for you that you can use in your filtering. Like, for example, again on the variant level, how many have called this variant, the quality, this indicator of the quality of the calls here, and some basic population level statistics that you get. For example, allele frequencies. This is actually, on UK Biobank, that now that you have, more than 500,000 whole exons, the allele frequencies is something that you want to look at, and this is actually an indicator you can discover like a lot of rare, variants that primarily, traditionally we don't have it in 1,000 genomes data sets, etc. Again, what you're doing here, you're using basically the same tools, the same calculation, but now it is distributed among multiple machines. And keep in mind, especially when you're working in the cloud environment, it is elastic. So if you want an analysis to be done faster, we have recommended ways of setting up your cluster that you can find in the documentation. So you can really, in a cost-efficient way, you can scale up your workload. So you don't have to wait necessarily for a night or something for analysis to be done. You just throw cores at it, and you can get your results in a matter of minutes, or depending on how you want to approach this, how performance is important to you. So as an example here to, looking at this, getting a summary statistic from your variants, I'm getting, I'm calling that function, but also, as you see, allele frequencies or some others are actually into more complex structures. They are like arrays that, let's say I want to have access to the particular element in the array. And this is a very simple, common API that you see in pandas or other places. You just specify by the index that you want to access. And now you're adding those columns here. So interestingly, keep in mind that you have all of this data. You have, I'm going back to the example of UK Biobank, for example. You have like 500,000 samples. But when you're doing on a variant level, you are doing calculating summary statistics. So the aggregation, you need a lot of machine horsepower to do the aggregation and analysis. But the table that you get the results. So this would be a table, with limited number of columns, and maybe 10 million or whatever the number of variants that you have. And if you have of order of millions rows and not really too wide tables, this is something like what we say we can collect it. So by collect, when on a Spark data frame, when you collect, all of these bits and bobs of data that are distributed among multiple machines, they are collected into one machine. And then you can write it into a pandas data frame or a standard CSV file, even one file. So if when you are doing this kind of workload, then at the end this is an example that I'm doing the analysis here. I need the scale, but then I want to go back and I'm a pandas user, for example. I want to collect that and just start doing my plotting and everything in my own way. You can easily do that. Simply, in this case, even I'm taking a random subsample, and what I do is that I change this into a pandas data frame. So simply, I call two pandas to it, and now I can just do some look at scatter plot. In this case, like what are the patterns of homozygosity and allele frequencies? Clearly we are expecting something from the theory, but this is interesting to look at how it is done. And then you can just combine that with the rest of the Python or R ecosystem. Or even, if you want to do anything ML, I don't know, if you want to do, for example, outlier detection, classification of points, k-means, UMAP, dimensionality reduction, you name it. Everything, then you have access to that ecosystem within the same platform. As I mentioned, even if I create this as a temporary view, I can access the same data set within the R runtime. So in this case, I'm selecting just number of homozygous and allele frequencies, and I can now continue an analysis in R. So the value here is, again, I go back to my own example. I preferred for a lot of data engineering and manipulation. I did a lot of work in Python, and I used to write my results as a CSV file, and then I would import those results into R, because for some, like R is actually really powerful for a lot of statistical functionality. But this case, I don't have to do any I/O. I do my work in Python, and then I can register that as a temporary view and access that within R or SQL, or even if you are more of a data engineer persona, you can do all of this in Scala, actually, which is the base of this work. Again, as another example of a built-in function, statistical genetics specifically function is a calculating Hardy-Weinberg. As you see, I can pass that again as a simple SQL, and it gives me the Hardy-Weinberg test per variant, where you can use to filter for your variants. The next step is obviously the more involved statistical genetics analysis, which is using whole genome regression with GLOW-GR, which my colleague Kiawesh will be walking you through it. And thank you so much. I would be happy to answer any questions. If you have anything, you can ask me on the Q&A session. Thank you.
Okay, thank you very much, Amir. I'm just going to share my screen again. So we're going to take a break now. So we're a little bit ahead of schedule. So I suggest that we take a break now until 11:30 or 2:30 on Eastern time, and then we'll do the final workshop, which is some really exciting work on doing whole genome regression on a massive scale that Kiawesh will walk through. And then we'll do some Q&A at the end. And there are some questions about how to run these jobs on the community edition. So we will tackle some troubleshooting as well at the end. But I would say for now, just relax and watch the demos in real time, and then we'll help troubleshoot at the end and answer all of your questions. So we'll see you back again in about half an hour. Thanks a lot, everyone. Okay, we're about to get started again. So I'd like to introduce Kiavash Kiamfar, who's a software engineer at Databricks. And he's going to be leading the third workshop, which is going to be describing GlowGR, which can be used for ultra scalable whole genome regression. Over to you, Kiavash.
Thank you, Will. Okay. So, hello, everyone. Thank you very much for being with us till this last session. I'm very excited today to talk about a very exciting feature that was recently added to the Glow open source project and Databricks unified analytic platforms for genomics, and that is what we refer to as GlowGR, which is an ultra scalable whole genome regression method. This was developed in collaboration Databricks, our team here in Databricks with Regeneron Genetics Center. And it's really a revolution in the way that whole genome regression based GWAS or genome-wide analysis studies are done in terms of the scalability as a speed, and we are very excited about that. So let's go ahead and dive into this. I think if you have worked in the genomic or statistical genetics workspace, you are familiar with genome-wide analysis studies, and we can put it this way, that GWAS saves lives and money. Genome-wide analysis studies are essentially a study of association of genome-wide variants with different traits, or what we will refer to as phenotypes also in this talk, in species. And in particular, diseases are considered one of the most important traits that are under a study in this kind of analysis. And as a result, the outcome of GWAS analysis can become very useful in healthcare. So what I mean by that is that let's assume that out of a GWAS analysis, we find out that some variant or a collection of variants are causing a particular disease. Therefore, based on that, then the drug developers can look for and develop chemical compounds that target that variants to essentially prevent that disease to happen. So as a result, this can be the underlying study that drives the development of drugs. An example of that is this study that was published in New England Journal of Medicine by our colleagues in Regeneron a while back, where they have found, based on this kind of analysis, a protein truncating variant that can help in curing chronic liver disease. And that has become the basis of developing particular drugs for this purpose. So, and then at the same time, we can go from drug discovery to, of course, personalized diagnosis and therapy, because these are all based on the genomic profile and the variants that drive the diseases, and therefore development of therapies for them. And another example is usage of the genetic variants as proxies for different kinds of interventions. As William mentioned at the beginning of this workshop, there are nice studies that you can do essentially this analysis in silico over the data that is available and come up with, instead of doing the actual intervention in a clinical trial form, first do it in silico based on the genetic profile of the population that you have, and then based on that decide whether a clinical trial is worthwhile to do or not. So an example of that, as William talked about, was the combination therapy that was done, that was discovered based on the Mendelian randomization framework using this GWAS analysis that pointed to reduced risk of coronary heart disease. And on the flip side of the cone, there are this can be used to save money by avoiding doing clinical trials that are gonna fail. And an example of that was the selenium supplementation that William talked about at the beginning of this talk. And if the genetic variants had been used and instead of the actual intervention, then it could have been concluded much earlier without doing the costly clinical trial that this doesn't have any relationship with the prostate cancer, and the clinical trial that had cost more than $100 million would have been avoided. So I think this points to the fact that the GWAS is the game of the future, because the more data becomes available about larger population with and the more precise this data becomes, the power of this kind of a study becomes more and more tangible, and we are gonna find very exciting results out of this in the healthcare domain. But on the other side, doing population scale GWAS is hard, in the sense that this is a complicated problem that are supposed to happen, that are solved over very large data. Biobank scale data on genotypic and phenotypic dimensions is just expanding in different biobanks. There are currently hundreds of thousands of samples in these biobanks, and of course, for example, for human genome, millions of different kind of variants are of interest to be studied for association. So this problem has been notoriously computationally expensive, and over the last decade or so, the methodology and algorithms to solve this problem has also evolved. It started with simple linear and logistic regression, then linear mixed models were used, and they were expanded into whole genome regression methodologies. And before the work that we're going to talk about today, the well-known methods like Bolt LMM and or SAGE or fast GWA are methods that have been used for quantitative and binary phenotypes, genome-wide association studies based on the whole genome regression idea. But why are we excited about GLOW-GR? Because this is a revolutionary method in terms of doing population scale GWAS. And why is that? This the algorithm behind GLOW-GR is based on the Regini algorithm, which was recently introduced by Regeneron Genetics Center, and you can read more about the details of it in this publication that is linked here on bioRxiv. But what is the difference of Regini with the previous algorithm that has been used is that Regini uses a structure that lends itself into parallelization very naturally. So the previous algorithms that are used in Bolt or Sage are in the form that you cannot get your large matrix of phenotypes and genotypes and chop it off into smaller pieces and do computation on it, and then merge the results of this computation based on proper linear algebra. Regini has this in mind from the very big inception of the algorithm, and that is what it makes it very powerful and scalable. So GlowGR is the parallelized version of Regini implemented on Apache Spark. And the reason we are very excited about is that although the Regini algorithm is in nature very parallelizable, and so on a single node machine, it is definitely faster than the previous methods, with with concordance in result without losing any accuracy or power of the tests. But it is limited to a single node machine based on the nature of its C++ implementation, C++ based implementation. And as a result, the maximum parallelization you can get out of it is probably the number of cores that you have in a single node computer. But GlowGR gets that the idea of that algorithm and implements it on Apache Spark. And if you are familiar with the Spark, it's kind of the de facto standard for implementation of parallelized processing over virtually unlimited in size clusters, and is being used by all the Databricks customers and many other organizations across the world to process just terabytes and petabytes of data every day and every year. Therefore, having a parallelizable algorithm and the power of Apache Spark, GlowGR essentially joins them inside Project Glow and makes that parallelizable algorithm really scalable virtually with any bound. The more nodes in the cluster you have, the larger the scale you can achieve in your study. And that's why we are very excited about it, because it's virtually the first method that lends itself very naturally to the expansion to the GWAS scales that we always wanted to do. So as mentioned, this is included in Project Glow. If you refer to the Project Glow website, as it was shown before in this slide, you can read more about it in the documents page, and also look at the code if you are technically interested. And at the same time, because Project Glow is part of the Databricks Genomic Runtime, part of the Databricks Unified Analytic Platform for Genomics, essentially, this is naturally included in the Databricks product as well. Just to give you an idea of the speed and scalability that this method provides, you can look at this from the perspective of this chart on the right. Essentially, on this chart, you can see the results on the running time of a UK BB scale study on 50 phenotypes in this biobank, and you can see here the comparison between Bolt, FastGWA, Regini itself, and then the GlowGR method. Number of cores is on a log scale axis, x-axis, and the running time is a log scale axis, y-axis. And as you can see, because for both FastGWA and Regini, you are limited to a single machine, the maximum number of cores you probably can get out of this is a handful, maybe around 16. And as you can see, Regini already beats Bolt and FastGWA orders of magnitude, because this is a log scale, and the amount of time reduces to something between 10 to 100 hours. But when but you are just limited to this in one single core. What GlowGR does is that takes that algorithm, and then you can just throw more nodes and workers at it and scale as much as you want. So there is no limit here. So as you can see, by adding more nodes to your clusters, you can get to a place that you can do this study even within an hour, which is quite exciting. And this happens at no cost in terms of accuracy. If you refer to Regini paper, you can see that the results of the Regini completely match what you can get at FastGWA and Bolt or Sage in terms of test powers and the variants that become significant at the end of your study. And GlowGR uses the same algorithm, so the results have the same power. Some other advantages of GlowGR before I go into the details of how it's implemented. It's both. It supports both quantitative and binary phenotypes. It's naturally and inherently has instrumentation to deal with rare variants when the case control ratio is quite unbalanced. And this comes from the nature of the FERT approximate FERT algorithm that is included in Regini and also in GlowGR. And it is designed to take care of the population structure and linkage disequilibrium based on the nature of the algorithm again. And what is very interesting about it is that the method, as I will show, is in 2 steps, and the getting the phenotypic representer or prediction that captures the essentially captures the polygenic effect across the whole genome is decoupled from the association test that you do subsequently to get the let's say your significant signals. Therefore, in the second step, you can use any test of that you like based on your preference and or the setting of your study based on the results that you have from the first step. So this lends itself to a lot of theoretical and as well as the computational and tool usage flexibility when you use GlowGR. And because it's developed in a Spark environment and can be very easily integrated with any other tool. So that's what you see in Databricks platform as well. You can easily use the data preparation or variant manipulation things that we talked about before in the previous workshops and get the results out into the GlowGR the workflow, and then at the end get the results and use all the visualization tools that are available in the data science world to visualize your results. So this is very interesting because all of this can be done on very large scale data seamlessly, essentially, which is truly unique to this Glow and Databricks platform in that sense. Now, So I hope this has convinced you that this is an exciting development. So I just want to go into a next phase of this talk and talk a little bit about how GlowGR works. So I don't want to delve into the mathematics and the statistics and linear algebra inside this model, but I just want to give you a very high view level of what gives this method its power, and where this difference comes from. Of course, for more detail, I'll refer you to the Regini paper and also our documentation on Glow website to get the details. But if, as I said, GlowGR happens in 2 steps. So step 1, we start essentially with a model, just the common infinitesimal model for genome-wide as the whole genome regression, where your model, your phenotype vector with essentially as a linear function of your covariates and your genotypes. The problem and your problem is essentially about estimating the coefficients that are given to these vectors, genotype vectors, and covariate vectors. GlowGR method is designed to work on multiple phenotypes at once as well. But this model, of course, being a statistical representation here, talks about a single phenotype. But this doesn't mean that this is just a single phenotype at a time. Method in computation. These operations that I'm talking about can happen in parallel over several phenotypes, as I showed also before in that performance chart. So the problem that arises in terms of scalability is this matrix X0, which is essentially has equal to the number of your samples, which is in the biobank scales is hundreds of thousands or even millions of samples. And then in terms of the variance, if you start the method with your genotype array, probably that's hundreds of thousands of SNPs, for example, in the case of UKBB. And therefore, this cannot be linear algebra on this size. It's just computationally prohibitive, and it gets a long time. Therefore, the GlowGR method does a reduction of the size of this matrix based on the statistical algorithm and the linear algebra under the hood. So we will have a step that uses essentially, instead of working directly with this matrix, we make some estimation of at this level, but by blocking this matrix into smaller blocks and running, fitting a ridge regression model over these blocks, and then getting my estimates of this vector or inside the blocks, not inside the whole matrix. And based on that, come up with estimates for each one vector estimate for which block, and getting that into a smaller model. So if you are familiar with ridge regression, the ridge regression has a parameter which is called the shrinkage parameter. And so in this method, on each block, we use a set of the shrinkage parameters that we refer to as alpha parameters in our documentation and code. So every block essentially becomes gets an estimate equal to the number of these shrinkage parameters estimates. So what happens is that instead of the number of variants being in hundreds of thousands or millions, the number of your essentially reduced genotype metrics, which become and the number of columns there becomes equal to the number of your variant blocks times number of your shrinkage parameters. And these shrinkage parameters, we just don't use one single shrinkage parameter because we want the size of the variants that have impact inside each block is unknown beforehand. So we use a range of shrinkage parameters to be able to capture any scenario that may happen inside the block. So as a result, the number of columns in this matrix becomes much smaller, from hundreds of thousands, millions to probably thousands. And then at the same time, the number of rows not shown here, but this operation can be done also, can be split over on the number of rows as well. So when we are doing this operation, actually in reality, we do this over blocks of rows as well. So it's also lends itself to parallelization on the other dimension. So this is smaller. Now I have this smaller model. Then out of this, again, another round of ridge regression happens to essentially replace this the term with an estimation, which is single vector estimation for the polygenic effect. This is done again by ridge regression with different values of the parameters. That regression is, in case of the quantitative variables, is quantitative traits, is a ridge linear regression, and in terms of and in case of the binary traits, is a ridge logistic regression. The difference here is that now I go ahead and find the best fit that I can out of the all different shrinkage parameters, so that it gives me one single essentially predictor for the whole genome for this particular phenotype. This is done also in a loco, which stands for leave one chromosome out scheme, so that the prediction is calculated based on essentially the fit that comes out of every chromosome other than the one that the SNP of interest is being is on, so that we can kind of avoid the proximal contamination in this method. So this concludes step one, and then this vector is then used to capture the polygenic effect in step two, which is the regular GWAS association test using linear or logistic regression. So that in this model, this is essentially the single genotype that you're testing against to see if it has a association with your phenotype. Covariates have their location, and instead of the whole genome infinitesimal model, you just capture, you just use this predictor that we have. And as you can see in the terminology of the linear models, this is essentially an offset term, because I don't have any coefficient for it. The coefficient is 1. So essentially what happens is that a step one generates a term. That term becomes an offset that is used in a regular linear or logistic regression in a step two. Here, of course, the linear regression term is shown. If it was logistic regression, it would be a sigmoid of this term. So that's about the theory at a very high level, but of course there is a lot more details about this, which you can look at in the Regini paper. So now I want to switch to the notebook and show you at a high level that how this works using Glow WGR functionality that is inside the Databricks or Project Glow. So again, this notebook is on for quantitative phenotypes, but where we have a very similar notebook also for binary phenotypes available on GlowGR website. You can refer to that in the documentation website on GlowGR and read about it. At the end of the page, you can find these two, the quantitative trait and the binary trait notebooks if you are interested to work on them. So back to my demo here. So essentially, what is the input to GlowGR? Input to GlowGR is your genotype data, it's your phenotype data, and it's your covariate data. So you will have some input preparation, which is essentially done in the Spark SQL ecosystem, and also using pandas data frames. And this, which I will talk about this in a little bit more detail momentarily. These inputs go into a step one that we just talked about. Out comes a phenotypic predictors that I was talking about, which is essentially that y-hat loco that we talked about, referred to here by y-hat data frame. And then this output goes into a step two, which is essentially a genome-wide association study using linear or logistic regression and using this predictor y-hat as the offset in these regressions. So, so let's go ahead and look at how data preparation and then each one of these steps is done. So we and in this cell we have imported some of the libraries that we need, essentially some functionalities from Glow and a linear model package from Glow, which is which contains the WGR functions that that we use, and then PySpark SQL functions and NumPy and Pandas for doing the data frame and linear algebra, little linear algebra that I will do here. So at the data preparation, at the data preparation step, we first I've defined some path to read my data. Essentially, here I have my variant data, which I will convert to a genotype call data in a delta table. This can be also read from a VCF or BGN, of course, but if you have stored your variants before in a delta table, just the most efficient to read them. Phenotypes, we have stored them in a tab separated or comma separated file, so that every row is essentially a sample and every column is a phenotype. So your phenotypes don't have to be just a single phenotype. So you can have multiple phenotypes on different columns of this file. And same for the covariates. If you have covariates from your PCA analysis, you can include them in here as well. And then I would like to, after the first step is done, I would like to save the outcomes of the first step for future use into a file. So this is the path for that file. So this is that decoupling that I was talking about, that when this is done, this comes out, you can save this output and then use it later for any other GWAS association that you do for these phenotypes, if you see proper on different sets of SNPs and anything like that. So that's why I'm interested to store this, and this is the fact that essentially this part of the work needs to be done just once, and then the rest, and the second step can be done multiple times. So and this also makes quite an impact in the amount of time that you need to do your whole analysis, because in the other algorithms before Regini, you every time you want to do you want to do the whole genome regression, you have to do everything from the beginning to the end again, while here just the first part is done once, and then you just repeat the second part whenever you have new questions about your new sets of SNPs or anything like that. So in the next step, we have to prepare genotypes, phenotypes, and covariates. So essentially here I have read in the first, what we need to do is this. We have to read our variants from our data tables. We need to make sure that our variants are split to biallelic variants as it's normally done in a GWAS analysis. And then I have to convert my genotype calls to integer numbers so that essentially I have an array of values for my genotype states, because that's the matrix, the genotype matrix that goes into the algorithm. And one thing that we do, if there is any missing genotypes, we as recommended in the Regini as well, they are replaced by the average of the other samples in that locus. Therefore, using this mean substitute functions that we have implemented in WGR library. And at the same time, if some of the variants don't show any variation across all your samples, you just want to filter them out. So this all happens in these few lines here. So the first line loads the variants, and then we have a transformer in GLOW, which is called a split multi-allelic. So the Spark data frame that is the results of this reading is fed into this transformer, and the result would be just another data frame which has biallelic variants. Then I need to add a column called values to my data frame, which is essentially the genotype states of the genotype column in my VCF, for example, or the table that table of variants that I read from the delta, and at the same time mean substitute it for any missing genotype as well. And then the last line filters all the variants that are just homozygous across all samples. If you look at the outcome of this operation, you see essentially a table with a schema similar to when we read a VCF, but with the added column value at the end. So you have contig name, start, and end, as it was shown by Amir in the previous workshop. The genotypes column is here, of course, with the which is a structure of, in my example, I have 100 samples just for the demo. And I have converted this genotypes column to a values array, essentially, where every number is the number of alternative alleles in that particular sample. So most of these samples are just reference allele. For this sample has had one alternate allele, and so on and so forth. So this is ready for step one now. The other two smaller steps is preparing the phenotype. So you need to read them, and then you have to make sure there are no missing values. And then they should be also standardized with mean 0 and unit standard deviation. And this operation is done here. Phenotypes are read as pandas data frame, not a Spark data frame. So I'm using read CSV function, which is in pandas, and it index on a column called sample ID, and then we essentially replace the missing values with the mean of the just like mean of the vector, as it was done for the genotypes as well. And at the next step, we are doing just a standardization, subtracting the mean, dividing by a standard deviation. And my file has several phenotypes here. I'm just focusing on 2 phenotypes for the sake of the demo. So if you look at what you have loaded, your pandas data frame looks like this: index of sample ID, and there are 2 trays, and these are the values of your continuous or quantitative phenotype. And then next step is covariates. What happens to covariates is exactly like what happens to phenotypes. I have 6 covariates here, for example. These are red. Missing values are filled with the mean, and then I am just doing a standardization here. A standardization is recommended, as it's recommended in the Regini paper as well, to avoid any issues in terms of interpreting the effect sizes at the end of the study. So now the next step is starting step 1 of GLOW-GR. So what happens? So as we talked about, GLOW-GR is a parallelized method over blocks. So the first thing that happens is that the huge matrix of genotypes has to be split into blocks across both axes. So this is what happens using a function which is called block variance and samples. And at the same time, we also, for this function, we need to extract the sample IDs and pass it to this function as well, because those sample IDs becomes the keys essentially to join our data frames during the operations that are done under the hood. So essentially at this scale, you have the genotype. At this step, we have the genotype data. We apply this function, and what we get out of it is a block genotype matrix, which we will refer to as block DF, block data frame, and another Python dictionary, which is essentially your sample blocks. What is sample or a sample block is essentially a dictionary that maps every block to the samples that are inside it, and we need to keep the track of this in the rest of the flow because the functions will need it. This is just a visual of what essentially this function does. So you have that big X0 function that we talked about on our slide. Rows are samples, columns are variants, and this is the conceptual imagination of that. If you block things, you will have then much smaller blocks. They are identified by chromosome and block number, and then block numbers across the rows as well. And the way this is represented in this Spark data frame is essentially that you have the identifiers for the sample block and header block, and in each row you essentially store the transpose of this matrix here, either in a form of a dense or a Spark storage. So how is this done? This is done by first getting the sample IDs out of your genotype data frame, out of your variant data frame. This is done by get sample IDs, which is a function in GLOW. And then I need to set the number of blocks and the number of blocks and my sample blocks and the number of variants that I want to have inside each block across essentially my columns. So here I have a pretty small example. I have the number of blocks is just set as 10, for example, and the variance per block are equal to 1000 variants. So these values are set, and this function that I was talking about, which is block variance and samples, gets the input of which are variant data frame, your sample IDs that you extracted from your data frame, and these two parameters, and essentially does this blocking, and the output will be essentially these two entities that I just talked about, which after it is run, if you want to have a look at it, you can see that you have a header that identifies each row, and that header essentially that row is essentially a variant in this block, which is the header block, and you have the sample block for that row, and you have the size of the essentially the samples that are in that block, and then you have the mu and sigma of the genotypes that are in that particular block. These values are used by the algorithm to do the bridge regression and reduction. You can also look at the sample blocks, which is essentially that dictionary that I talked about. The sample block 1 includes this sample, so on and so forth. Next step within step 1 is to do that dimensionality reduction that we talked about, which is done by the function ridge reducer by the class ridge reducer, which has a couple of functions that I'll talk about. So the input here is essentially your blocked genotype matrix, your sample block mapping, the phenotype data, and the covariate data. These go into a fitting function in this ridge reducer class, then the output would be a model data frame that has essentially the entities of a model, and then after that the transformation is done on your phenotypes on this set of inputs to get essentially the estimates for that smaller, which present that smaller, the reduced matrix that I was talking about. You can combine these two functions by the function fit transform as well. And that is how this is done. So you essentially, you get block label df, which is your phenotypes essentially, and sample blocks and covariate the data frame. And what comes out of this is essentially a reduced block data frame, that instead of having one row for each variance, now you have essentially one row for each block and shrinkage value combination, and these are the values. The values are essentially the values of the vector. The next step, you get the results of this and do the ridge regression or logistic regression, depending on whether you are doing the quantitative trait or binary trace. And this is done in a leave one chromosome out scheme. So again, here I have all the previous input except that the block block genotype matrix is replaced by reduced block genotype matrix. And what happens is that this fitting, you have to do this fitting. Out of the fitting comes the model data frame, just like before, but in terms of a schema, but you also have a cross-validation data frame that essentially tells you which one of these models is gives you the best fit. And out of that, then depending on whether you're doing binary phenotypes or quantitative phenotypes, you feed the results, you use this model and cross-validation data frame and pass it into this function to get the what we wanted out of step one, which was the phenotypic that y hat local vector. So when you do this, how does this happen? So here I'm working on quantitative traits, so I'm using the ridge regression class. I initiate the class. The initiation of the class essentially creates these alpha values, which are my shrinkage parameters in ridge regression, and I use the fit function of this class and give the inputs that we mentioned above, and I get the model and cross-validation data frame. And having those, I fit them into a transform local function, and the result will be my y hat data frame, which essentially are what I wanted out of step one. And they look like this. Then essentially for each sample ID and for each contig name, I have for any phenotype, I have a number. And because this is a local scheme, you can see that I have different numbers if I have for contig name 21 and contig name 22 for the two different chromosomes. I save this output into a CSV or into a delta table or any medium that I like for future use for the step two, which can happen at any time or can be repeatedly happen. Step two is essentially just doing linear regression or logistic regression, depending on whether your phenotypes are binary or quantitative. In this case, I'm talking about quantitative phenotypes. So the meaning of having quantitative phenotypes is that just offset means that you can subtract your essentially phenotype. You can get this vector, which is supposed to be an offset, the y hat df, and subtract it from your phenotype vector, your y vector, and that's just takes care of this offsetting fact. So this is what essentially we are doing here. I have the phenotype vector. This vector has come out, so I do this subtraction. And in order to use this with the linear regression function that we have in GLOW, we have to essentially reshape the data frame, and we have a function which is called reshape for GWAS that does that. Out of this comes the data frame, which is essentially and we refer to as adjusted phenotypes because that offset has been subtracted from it. Then the last step is essentially just doing the linear regression, and for that we have a very simple function in GLOW, which essentially gets your variants and gets your phenotypes and gets your covariates and does the linear regression in parallel mode for you. So essentially for any combination of your phenotype and every one of your SNPs does the test for you with the correction that has come out of WGR and gives out the statistic regarded to that test, essentially the impact, the coefficient beta, the standard error of that coefficient, and the p-value, which shows the significance of that coefficient. And you can have this data frame and store it as a delta table, as we have done here. And this would be the result of GWAS analysis, like any other GWAS analysis you do. And after this, it's just up to the user how to use it. You can do the visualization and draw a Manhattan plot, as we have done this with this package here, or any other visualization scheme that you want. So I'm gonna go ahead and stop at this point and hand this back to William. I would be happy to answer any questions that you may have. Thank you very much for listening to me, and I hope you use GLOW-GR in your practice and find it just amazing. Thank you.
Okay. Thank you very much, Kiavash, for walking through how to use GLOGR for whole genome regression. As a next step, please sign up for a free trial or start a proof of concept by following the link databricks.com/genomics. And also, please download our ebook on real world evidence, dbricks.co/ebook RWE. And in a month's time, we'll be hosting another workshop on broadening this out beyond genomics to healthcare in general, and how you can use Databricks to do healthcare analysis with diverse data sets such as real world evidence. And I'd like to thank the panelists today, Kiavash, Amir, Henry, and Karen, and also Evan Maxwell from the Regeneron Genetics Center. We started this collaboration with Regeneron about 3 years ago, and it's just been amazing to see the progress that's been made. We've learned a ton from them and from our other customers, and we've open sourced this work as part of Project Glow. So please also check out projectglow.io. And with that, I think we've got a few minutes left, so we can go through some of the questions. There's been a bunch of questions in the chat throughout the session, and if I can find it, we'll go through a few questions and then we'll wrap. How do I show the? Okay, I'm going to stop sharing and then go to the Q&A section. Okay, so looking through the questions. Kiavash, there was a question about GLOGR. So is the blocking of X0 determined from the kinship matrix population structure or the LD blocks? So is the blocking of X0 determined from the kinship matrix or population structure?
Actually, the blocking is mostly the function of the computational burden that it causes. So we would like to keep the number of blocks and number of variants in each block such that the computation that happens for each block is controlled and doesn't the algorithm doesn't come bogged down by that. At the same time, we don't want to have very small blocks so that the statistical meaning of the block is lost, and if the block gets very large, then we are just back to square one in terms of losing the capability in terms of parallelization. So we have recommendations regarding this case by case, and some of that is also available on the GLODoc. So this depends on the problem actually. But in regard to LD and the population structure, that has less of an effect on choosing the block size, because the nature of the algorithm, if people read about it, essentially takes care of those issues inside the blocks, at least inside the blocks. There are some concerns about the intra-block the relationship, which are also addressed in the Regini paper.
Yep, I want to add to that, as Kiavash said, the main value proposition for this method also is that automatically it corrects for LD and kinship. By training an initial model under the null hypothesis of no association. So even you can mix multiple ethnicity and admixture, and mix individuals in the same GWAS, and the initial step corrects for that.
Cool, thanks, Amir. And following up on that, are these genome-wide association studies limited to single phenotype or univariate analysis, or are you capable of performing multivariate GWAS, multiple phenotypes simultaneously?
Yeah, the second is true. As I mentioned through the talk as well, no, it's not limited. It can be done for multiple phenotypes simultaneously. Even the example that I showed I had actually 2 in this small demo, but it can be done on multiple phenotypes in parallel at the same time, and that's actually a powerful feature of this method that we are quite excited about.
Right. So that's one of the reasons why it's more computationally efficient, right? You can run multiple phenotypes simultaneously.
Yeah, that's one
and
of the reasons.
That's
yes,
why you would use GlowGR.
Yeah.
If you have multiple phenotypes. Yeah. Awesome. Okay. So there was also a question about, I'm going to throw this at Amir, about earlier in the session, Karen showed that some of the variants had been annotated with VEP or SNP-F. How do we do that? And how do you also use your own bioinformatics tools on the Databricks platform?
Yeah, absolutely. I will pass the annotation piece to Kialash, since he also wrote the parser for ingesting custom annotations. But the second question, which is about customization, actually, this is one of there are 2 ways that you can really leverage the platform and the Glow's capabilities. Think about it as you can scale existing bioinformatics pipelines using Spark and Glow. For example, as one example is, Glow that we have is an API called the Pipe API. If you have a VCF file and you ingest it using Glow, and let's say you have a workload, you have a, let's say, shell script or any other application that takes a VCF as an input, you simply can pass the name of that, the path to that script and the parameters of the script, and call the Pipe API on the data frame that has the VCF files. This, under the hood, it distributes that workload among multiple machines. So traditionally, when you think about we are like bioinformatics, where HPC workers, we have this Q subs and everything to distribute that. This takes all of that complexity away. You even don't need to think about any of your resources. Just pass that Pipe API and the script path, and that distributes it. This is one way. There are multiple other ways that you can actually go lower level with Spark and distribute the task.
Thanks, Samir. Yeah, so I think the key point is that the distribution is largely abstracted away from the user. So you don't have to worry about managing Spark clusters or setting and configuring them. That's all taken care of for you. The other thing is that the back-end cloud storage and cloud compute is also largely abstracted away from the user. So you can run this kind of analysis in the cloud without needing cloud expertise. You don't have to be a DevOps engineer or an expert in cloud architecture to run any of this. It all just works as if you're running it on your laptop. And then it's a few lines of code to distribute it with Spark. Okay. It's another question probably aimed at Amir. Can you talk about how you integrate other data sources in Delta Lake, and what kind of data sources do we work with? So, for example, medical literature or electronic health records and things like that.
Exactly. So that's a great question, and that's primarily the main advantage that we are seeing when you're using this, framework, is that this is compatible. You're using the same APIs, as I mentioned during the demo. Use the same APIs that you use for analyzing other data sets on genomics data. For example, a good example, actually, we recently wrote, and we are releasing it as an open source project called similar, the Glow, and then this other one is called Smolder. And the idea for this one is that now we can ingest HL7 files. So HL7 messages, they are conventional, messages that you get in hospital systems. These are like, health level 7 data. So Smolder package, what it does, it can ingest that in stream or batch. And the same way that you are giving a structure to semi-structured VCF data, you're giving structure to HL7 messages, and you can add that to your Delta Lake. So now imagine you have electronic health records, they're coming in, on stream, you write it in a Delta Lake. You have genomics data in a Delta Lake. The same way you can ingest imaging data, or other like FHIR format, that's another EHR format. Now that you have all of these as Delta tables, which are basically based on Spark data frames, now you can just use let's say you want to join, you use Spark, the data frame join on the sample ID. Now that you have, annotation, let's say for example of GWAS, example of 1,000 genomes, you have this metadata associated with samples. Imagine that you have a table, you join the sample table that you have all that mixture information or ethnicity information with their EHR information that you have extracted from other places. Now you have this enriched phenotypic table, for example, that you can do GWAS. Think about it like, or if you have imaging data, you extracted features from the images, you can do the same thing to merge and join.
Right. So Delta Lake adds structure to semi-structured and unstructured data sources like electronic health records and imaging data, and so it allows you to build more robust pipelines. Okay, we have a few more questions coming in. So Kiavash, before you do step 1 and step 2 of FlowGR, how do you prepare the data? Do you start with raw genotype data, or do you have to do quality control or imputation? How do you prepare the data to get into FlowGR?
Yeah, that part is just separate from this methodology. So any kind of quality control and imputation that is needed to be done is assumed to be happened before we get to this step. So there's no difference in that term with any other GWAS analysis.
Right, and the quality control is, those steps are also built into Glow, but separate.
Yeah. Amir talked about them. The only thing is that the LD pruning and stuff like that doesn't need to be included, because as Amir mentioned, and I also talked about it in the presentation, there is no need that is taken care of inherently in the structure of this algorithm.
Right, so the algorithm radically simplifies the whole process, because you don't have to do a separate LD pruning, you don't have to do separate principal component analysis. That's all taken care of with this new method. Is that right, Amir?
Yeah, for peer principal component analysis, probably if you want to use them as covariates, that needs to be included. Actually, Regini method includes the PCAs that are in the UKBB as the covariates in this method.
Yeah, I just wanted to mention that you need still to have the PCAs at the end of it. But for example, kinship, the cryptic variation, cryptic relatedness that you see in samples, like usually you have the initial step in GWAS, you make sure that they are unrelated. So the whole genome regression methods, this is like SAGE is the original one that had it. You take kinship matrix as a covariate in your analysis under it, but Regini streamlined that.
Got it. Okay, and that's particularly useful when you get up to scale and you have cryptic relatedness between samples. Awesome. Yeah, so in fact, there was another question from the same person, diving a little bit more into the details. So does changing the sample block size, the genotype block size, does that influence the results at all? And do you have recommended values for these? Or is it context dependent?
I probably defer this to Karen, because she had done a little bit of analysis on this. So I don't know if.
Oh, there you are.
Yeah, so we've done testing with Regeneron when it comes to different block sizes. The recommended block sizes are actually set by default during Glow WGR. So, and that's been tested up to the 500. Sorry.
He doesn't like that.
It's not like Glow WGR apparently. But yeah, we can test up to 500K scale. With smaller sample sizes, you might want to change. I'm so sorry. You might want to change the block sizes, but this is what we've tested up to.
So missed that because of the barking. Well, what sample sizes you tested up to?
This is up to 500K. This is for UKBB.
And for joint genotyping, how far up are you going to go?
hope the dog survives that.
I'm sorry.
No, this is how the reality is. I'm about to have a baby crying behind me, so you're in a better place.
Yeah, I just tried to escape. For joint genotyping, we've tested, as I mentioned during the slides, up to the 50K whole exome scale. On the genome scale, we've tested up to, actually, Will, you might be able to refresh me better on this. Personally, I've tested it up to, I think, 10K whole genome samples.
Awesome. And actually, Karen, like at what stage would you consider moving over to Spark for this analysis? And then what stage would you use single node? This is for joint genotyping, GWAS, and QC.
Yeah, I mean, so you can run these processes on as few as, say, 5 samples. Although at that scale, it's probably not necessarily worth it because these are kind of the toy data sets that we've been playing with. For joint genotyping, we've been recommending it for users with more than, say, 1,000 samples or so, and for whole exome sequencing in the 10,000 sample scale.
Okay, well, I think we'll wrap up there. So the notebooks are freely available on the Glow docs, and we'll send out a recording over email and the slides, which also contain links to the notebooks, the ebooks, the signups, etc. And yeah, please let us know if you have any questions. And Kristen, is there anything else to add before we conclude?
Yeah, so I'm going to launch one more poll. We would love your feedback on this event. This really helps us continue to improve future workshops. So yeah, just a quick 2-question survey on how you thought this went. But yeah, that'd be really helpful. I'll keep this open for 1 more minute. Other than that, thanks for joining us.
And one more question is, who should people contact if they have questions after the event?
Yeah, I will be sending an email tomorrow with all of the links, and then you can reply directly to that email.
Awesome. And then you'll forward it to the relevant people.
Yep, definitely.
Okay, brilliant. Well, thanks a lot, Kristen.
Sounds good.
For organizing this.
Everyone.
And thanks to everyone for attending. Really appreciate those who stuck around to the end. And we look forward to hearing from all of you in the future.